Morning, everybody. Thank you for joining us for this NIEHS Council meeting. I am Kyle Walsh. I've introduced myself to many of you already, but for purposes of this, it's great to be here. I joined NIEHS in October, and so this will be my first council meeting with you all. Really excited to share with you exciting updates about what we're doing, and also really enthusiastic to get your feedback on how we can improve operations. So I want to start with the important things in the important red text. So pursuant to the government in the Sunshine Act, all aspects of this meeting are open to the public, except the review, discussion, and evaluation of grant applications and related information. So we're all clear with that. And we are recording and we'll be ready to go. So David's going to help lead us in some introductions, and then I'm going to start with a presentation. I'm going to do my best. We have a lot of slides and a lot of content and a lot of exciting updates to share, but I'd like to reserve, at least about 10 to 15 minutes for some questions at the end. And so as we're moving through things, if you think of questions, maybe make a mental note of that, and we can roll right into that when we continue through. So I'd like to get this show on the road. And so, David, can we do attendance and some introductions?
Yes, we'll start with letting the council members introduce themselves, and I'll just go down in alphabetical order. And let Dr. Cormier, if you'd like to introduce yourself.
Sure. Good morning. My name is Steph Cormier. I'm from Louisiana State University, and I am involved in the council, and I've been studying early effects on children for over 25 years. Specifically, we study environmental pollutants known as environmentally persistent free radicals, which are found in Superfund sites, and I study the intersection of those pollutants with respiratory viruses. Infants are my thing.
Thank you, Steph. Dr. Deschênes?
Morning, everyone. I'm Olivier Deschênes. I'm a professor of economics at University of California, Santa Barbara. I can report great to see you this morning. I haven't seen the sun quite yet. And my research is on the health effects of environmental policy and environmental factors. Great to be with all of you here today.
Thank you, Olivier. Dr. Greenemeyer?
Hi, I'm Tim Greenemeyer at the University of Pittsburgh. I'm a neurologist and neuroscientist, and mostly I study gene-environment interactions in neurodegeneration.
And you win the backdrop award today, I think, Tim. Dr. Hood.
Hello, yes, Tim, quite nice. I'm Darrell Hood, professor at The Ohio State University. I am an environmental public health neurotoxicologist, co-architect of the public health exposome framework, which falls under the exposomics umbrella. Glad to be here.
Thank you, Darrell. Dr. Hornbuckle.
Good morning. I'm Carrie Hornbuckle. I'm a professor of civil and environmental engineering at the University of Iowa. My research concerns air toxics, especially emissions from building materials and remediation of older buildings. I'm also the director of the Iowa Superfund Research Program. Nice to see you all.
Thank you, Carrie. Dr. Hoya.
Yes, I'm Catherine Hoya. I'm an epidemiologist. I'm a professor in the Department of Biological Sciences at NC State. I study epigenetic responses to environmental pollutants throughout the life course. Thank you.
Catherine. Dr. Mutlu.
Good morning, everyone. Good to be here today. I'm a pulmonary and critical care physician. I'm a professor of medicine and the chief of the section of pulmonary and critical care at the University of Chicago. My research focuses on understanding the mechanisms by which particulate matter air pollution causes cardiopulmonary disease, with a specific interest in the role of alveolar macrophages.
Thank you, Culkin. And last but not least of our full council members, Ms. Sebasta Kennedy.
Hi there, good morning. I'm Maria Sebasta Kennedy. I'm a clinical professor of law at UNC School of Law, where I teach environmental courses, including environmental justice. I'm the author of North Carolina's Environmental Practice Guide. And my students and I are looking at well water contamination in North Carolina. Almost a third of the folks here are on well water, and it's a particular problem in the rural areas. Good to see you all.
As one of those people on well water, I thank you for your work. And we do have 3 ex officio members representing other federal agencies to the council. So the first is Yulia Kara. Yulia, would you like to introduce yourself?
I am representing the Centers for Disease
Control. Your audio is very, very quiet, Yulia.
I'm representing the Centers for Control Disease and Prevention. Can you hear me now?
Yes, we can.
Oh, okay, great. Yes, I'm the Associate Director for Science at the Division for Environmental Health Science and Practice. Thank you.
And Dr. Fitzpatrick from the FDA?
Yes, Susie Fitzpatrick from the FDA Human Foods Program. I work on, I'm a regulatory toxicologist working on new approach methods and co-chairing ICVAM, which will be under NIEHS and will be going somewhere else soon, I think. Nice to be here, and thank you for the invitation.
Thank you, Susie. We have an ex officio from EPA who I don't believe is on yet. Maureen Gwynn, are you on?
No,
she may join us later. I think she was planning on joining today.
Yes, I'm still trying to connect with her. Sorry, David, I'm still trying to connect with her.
Okay, thanks. We do have one council member who is unable to join us today. That is Dr. Patricia Nez Henderson. She's a public member of council, so she was unavailable today. Next, I'll move on to members of the NIEHS senior leadership to introduce themselves, and we'll start with Dr. Archer.
Good morning, everyone. I'm Trevor Archer, the Deputy Director here at NIEHS. I'm a biochemist by training and with interest in environmental impacts on gene expression and epigenetics. It's good to have you here, and thanks again for doing this. We really appreciate it.
Thank you, Trevor. And Ms. Mathis?
Good morning, everyone. I'm Jay Ingrid Mathis. I'm the Associate Director for Management and the Executive Officer, overseeing business operations and scientific support services. Thanks for joining.
Thank you, Jay. And then I will also send regards for Doctors Zildin, Fessler, and Pattisall. They are all at an important meeting in Bethesda this morning and unable to join council today. But we do have representation from our intramural divisions on council normally. So with that, I think I get to move on to my fun bits of the meeting. So we'll start with some Zoom etiquette. So the members of the DERT senior leadership and council are all on a Teams meeting, but the rest of the participants are participating through the webcast. The participants in the webcast do not have the opportunity to raise their hand and speak, but if you have important feedback to share with us, please send an email to the NIEHS website or email address. That is national.advisory.council@niehs.nih.gov. And we will be monitoring that email address throughout the meeting to share any feedback from the public. For those members on the teams, please remember to keep your microphone muted unless you are called upon to speak. We are encouraging you to use the raise hand function to be acknowledged. Kyle and I will be monitoring that, and we'll call on you to speak as you raise your hand. For bandwidth purposes, it's helpful to keep your video turned off. A reminder that we will have a break this morning, and during that time, please turn your microphone and video off during the break. If you are on VPN, you may want to turn that off to preserve bandwidth. For council members in the closed session, we may need to absent you from the meeting briefly for any conflict of interest reasons. To do that, we will place you into a waiting room in the team, so you do not need to leave the team's meeting. We'll place you into the waiting room, have the discussion, and when we are done with that discussion, we'll bring you back into the team's meeting. We won't need to do that during the open session. We will be taking some votes both in the open and in the closed session. That voting will be conducted using the electronic council book. If there are any technical difficulties, we think we've gotten all of those worked out, but if there are, we'll resort to the old-fashioned raise hand voting in the teams, but hopefully the ECB will work. I will prompt you in my presentation when we're getting ready for the vote to log back into the electronic council book. You do have access to both Kyle's slides and mine in the ECB, or you should have access to that. Next, I get to read my fun boilerplate language addressing the review of confidentiality and conflict of interest. And I read from the quote, policies and procedures for preventing conflict of interest and confidentiality of grant materials are contained in the conflict of interest forms emailed to you by Ms. Valerie Bartlett. That states, this certifies that in the review of applications or projects conducted by the National Advisory Environmental Health Sciences Council on today's date, I absented myself and I did not participate in the discussion of nor vote on any applications or projects in which I, or to my knowledge, my spouse, minor child, or close professional associates have a financial interest, nor on any applications or projects from an organization or institution where I am an employee, consultant, officer, director, or trustee, am negotiating for employment, or otherwise have a financial interest. In council actions in which we voted on a block of applications or projects without discussing any individual one, the on-block actions, my vote did not apply to any applications from any institution fulfilling the criteria in the preceding paragraph. Members should sign the certification and email that back to Valerie today if you have not already done so. Maureen Gwynn has joined us. Maureen, if you would like to introduce yourself.
Yes, I'd be happy to. Thank you. Can you hear me?
Yes, we can.
Okay, great. Sorry to be late. I was on the phone and it wouldn't let me unmute. I am Maureen Gwynn. I am here as the ex officio member representing the Environmental Protection Agency. I am the acting associate administrator for the Office of Applied Science and Environmental Solutions and the Office of the Administrator at the EPA. Happy to be here. Sorry that I was running from one other meeting and I'm a little bit late. So thank you very much.
No worries, Maureen. We're glad to have you on.
Thanks.
So next item of business for council is the consideration of the minutes for our October meeting, which was held in September of 2025. Council members have been given access to the meeting minutes from our last meeting in September 2025 in the electronic council book. The meeting minutes can be found on the council information page on the agenda tab if you scroll down below the agenda itself. I hope that council members have had an opportunity to review the minutes. And is there any discussion from council of the minutes? If not, may I call for a motion to approve the minutes?
So moved.
Mm-hmm.
Thank you. That was Daryl Hood as a primary, and I didn't catch who the other one speaking was. So do I have a second of the motion?
Tim Greenemeyer.
Okay, thanks, Tim. And if I can then call for a vote, please vote in the electronic council book on to approve the minutes, disapprove the minutes, or abstain. Valerie or Ashley, if you can let me know when we have quorum.
Valerie, do you see the votes?
Not yet, I'm still looking.
I'm sorry, I'm having trouble figuring out how to vote. I can see the minutes, but I'm sorry, I can't see where the voting. Oh,
Same here. There's no place to vote.
You
Same.
click on the yes, and the little arrow will indicate that you voted yes.
Oh, got it. Thank you.
Did that work?
David, for this one, I'm not seeing the votes. Valerie, are you?
I see yes, but I can't select yes.
You can't select. Okay, let's just do a
Shall we
hand
just
vote
go with the
for
raise hand function?
minutes, and then we can try again with the SOP, because I can see the voting for that.
Okay. So all in favor, please use the raise hand function to raise your hand, please. Looks like that is quorum to approve. Are there any no votes? Assuming that those 5 hands are legacies. No votes, no votes, no abstentions. So then we can move on. And Kyle, I think that you are up to give your presentation.
All right. Get this in presenter mode for us. All right. So this is our official report to you. And I did kind of a brief introduction of myself. I wanted to put in an introductory slide. It got very weird putting bullet points in about me and where I've been. So I used some AI and created a map for you. So as a brief introduction, I did my undergraduate degrees in molecular genetics and physical anthropology at Ohio State. I did doctoral work in chronic disease epidemiology at Yale, where I completed my PhD at the Yale School of Public Health. I did a postdoc that was jointly between UCSF and UC Berkeley, and eventually joined the faculty in the Department of Neurosurgery and the Department of Epidemiology and Biostatistics at UCSF, where I ran a lab focused on largely brain tumor and cancer etiology, so childhood leukemias, malignant gliomas in adults. We had both a computational research space, as well as, did animal modeling, in vitro modeling, et cetera. And so really was working to kind of bring together both the sort of classic epi and, nested cohorts and case control studies work with doing functional genetics and functional toxicology work in the laboratory. About 8 years ago, I came to Duke, where I was a professor in the departments of neurosurgery, pediatrics, and population health sciences. I led our division of neuroepidemiology, as well as working within our cancer center, where I was the co-leader of the neuro-oncology program at Duke. I also worked a lot with the Alzheimer's Disease Center, and I led the R25 research training grant for our Duke and UNC Alzheimer's Disease Research Center that was a P30 funded by NIA. And as you know, in October, I didn't have to move very far, and I now get the immense privilege of coming to work every day at NIEHS. And so it's been a really wonderful transition, and I think things have gone quite smoothly, and a great deal of that is due to the incredible support of our extramural director, David, all of the staff, as well as our intramural folks, and Jay Mathis, who's also on this call and has really been a cornerstone of all of the sort of management pieces for a research institution that has, more than 1,000 employees between, our federal employees and our contractors. And so it's been wonderful, and I've really enjoyed it and enjoyed getting to go out and meet with folks from the Superfund Research Program, I'm going to be out at the Society of Toxicology meeting shortly, and just getting to know all of the folks who both work with NIEHS funding, but who do, incredible research that really moves the needle and pushes forward fantastic innovations that we also leverage here in the intramural program and in ICs, really NIH-wide. So I will move directly to some fun budget stuff. So this is an overview of funding summaries from 2023 to 2026. The sort of only thing that I want to sort of draw your attention to here is that with what the fiscal year 2026 enacted budget that was passed by Congress is it essentially is putting us at more or less a flat budget year over year for the past 3 years. There was some discussion around potentially seeing reduced funding for our Superfund allocation that, as you recall, does not come from the HHS and labor allocation, but rather from the Environment and Interior allocation. And that, was down a couple $1 million, but again, mostly in a flat funding landscape at this time. So I thought it would be helpful to talk a little bit about what's going on in terms of sort of HHS-wide, and then how that is being enacted both at the National Institutes of Health, as well as what we're doing here at NIEHS. And so the Make America Healthy Again Commission was assembled to advise the president on the chronic disease crisis, particularly among children. They have identified deliverables last year. So initially, there was an assessment report looking at the scope and the drivers of this chronic disease crisis, followed by a federal policy roadmap that's looking toward prevention of these diseases, particularly through alterations in regulation, which is more of an EPA and CDC and outside of the NIH framework. But as well as having these be really heavily research informed, which of course is certainly our wheelhouse here. The composition of this commission is it's a multi-agency federal leadership involvement. So at HHS, there's extensive representation from NIH, from FDA, as well as from CDC, and then outside of HHS. The USDA, the Environmental Protection Agency, as well as the Department of Education and others are extremely heavily involved. And ultimately, the goal here, I think, is a goal that everyone can get behind, which is to transform our health systems to both prevent and reverse chronic diseases, particularly chronic diseases of childhood. So in the commission report, identifying major drivers of childhood chronic disease, this won't be news for anyone who is NIEHS adjacent, and members of council are certainly much more than adjacent. They are deeply integrated and involved. But these major drivers that were identified and discussed was particularly poor diet with a focus on ultra-processed foods and high sugar foods. Sedentary and stressful lifestyles, including, increase in screen time, both television, computers, mobile phones, and related sleep deficits that come from some of that. And then, environmental chemical exposures, right, was really the third piece of this. And so, this includes everything from, herbicides and pesticides to micro nanoplastics to dioxins and other chemicals. And so this figure here, which is actually included in the report, highlights some of those particular areas of concern. And as I don't think we need to go through them all in detail, but I think you can appreciate that, heavy metals, air pollutants, pesticides and herbicides, EDCs, right? These are really places where NIEHS has and will continue to focus a great deal of our research enterprise. So the strategic framework, is not just looking at what the problem is, but really focused on reforming that. And so reforming this from the perspective of food, of health, and scientific systems that can identify what the underlying root causes of the chronic disease epidemic. And so, as an epidemiologist by training, and I'm very, very pleased with this idea of, wanting to get to etiology. And of course, we're all aware that the etiology can sort of be, it's obviously multifactorial, right? And it's going to be really important that we critically evaluate, the relative contributions as well as the interactions and how these things are modified by, environmental contaminants, their effects being modified by diet, behavior, nutrition, genetics, all of these things. And so HHS has, essentially mandated what will be an initiative on chronic disease to leverage and align both our existing NIH research projects and really to improve coordination on chronic disease research with the ultimate goal of generating actionable results. And so that's a priority and a highlight here, is that this is, really that research to action paradigm. And a lot of the work that both our intramural and extramural community at NIEHS do in terms of community engagement and those things really positions us nicely for this opportunity to really move research to action. So how are we managing all of the sort of things that have been highlighted and assigned and worked upon? As you all know, Dr. Wojcik has now moved from being the NIEHS and National Toxicology Program Director to being a senior advisor in the Office of the Director. And so he's a senior advisor to Dr. Bhattacharya at NIH, really advising and helping coordinate and strategize around NIH's Make America Healthy Again strategy. So really looking to advance an agenda that unites research and innovation across institutes and centers and partners with our other federal partners, including, the EPA and the CDC and others. Looking to identify root causes of chronic diseases, strengthen the nation's health resilience, and there's a large contingent and component of this that's really focused on data-driven prevention strategies and data harmonization across institutes and centers and agencies, because, again, everybody has sort of components of the full picture. And so it's going to be really an important data initiative to bring all of those components together for a unified research strategy. So ultimately, I think that we see this as a really incredible opportunity for NIEHS, both in terms of a scientific opportunity and a leadership opportunity for the Institute to align with this high-profile national initiative. And, really wonderfully, the strategic plan that was published in 2025 is already just incredibly well aligned with Make America Healthy Again priorities. And so it's going to be, I think, a great opportunity for NIEHS, NIEHS funded investigators, as well as our intramural folks to really be sort of top level contributors to this high profile initiative that I think we can really all get behind. I just wanted to provide an example of how we are sort of leveraging these opportunities. So there has been a recent investment in farm modernization and food supply security. This was announced jointly by the EPA, USDA, and HHS about 3 weeks ago on a Friday. And so what they announced is a coordinated federal strategy with a little more than $1 billion in investments to modernize agriculture, strengthen long-term food supply and security, and to understand sort of MAHA-related health issues that can be linked and or inextricably linked really from our food supply. And so major priorities that have come through that we will be working with NIH and the Office of the Director toward involve better understanding risks of agricultural chemicals to humans, expanded investments in regenerative and sustainable agricultural practices. That'll be the USDA side of things. And accelerating innovation in agricultural technology and farm modernization, which is going to be sort of jointly ARPA-H and the Environmental Protection Agency. And so for us, agricultural and chemical exposures and their health effects, aligns incredibly well with work that we've already done, with work that's ongoing, with work that we want to continue doing. And this additionally ties into occupational exposures, including impacts on agricultural workers and rural communities. And so there's of this $1 billion, there's $100 million that will be NIH separate from the ARPA-H piece, which is a different $100 million pot that will be supporting work in this arena. All right. So from the strategic plan and others, these are our continued areas of research emphasis. And I just thought that this graphic could be, a nice way. We're not going to go through each piece, but we'll use it to highlight some things we're excited to talk about and share. So the exposome, right? Rick was a major driver of pushing forward exposomics research. And I think that that's just fantastic. I, of course, am incredibly aligned with that. I trained in the lab that did the first genome-wide association study. And so anytime you can move from looking at one or two things to looking at, thousands or tens of thousands of things, as long as you are powered to do so and, interested in doing functional follow-up, I think, it's a great opportunity for, really disentangling these effects, especially when we think about things like chemical mixtures and, places where effects are not attributable to a single compound, a single toxicant working in isolation. So highlights from our Network for Exposomics in the United States, so NEXUS, the Center for Exposome Research Coordination that has been funded through a U24 mechanism has been doing incredible work catalyzing integration of exposure data across both molecular, clinical, and population level research, has been working to disseminate standardized analytical and computational methods, really supporting data science that will lead to reproducible exposome-wide association studies. And I think this is incredibly important. Again, not to belabor the point, but I remember, 12 years of my life where nobody could agree how to control for population stratification in a GWAS study, or what the sort of p-value threshold would be. And, we eventually got there and you get to a community of science understanding. But this framework is really going to be a nice way to accelerate that and solicit buy-in from all of the partners in this space. So establishing infrastructure, training programs, and community engagement platforms has been a major focus. And I think that this is really building on success of other projects and programs that NAEHS has supported, including CHEER and more recently the HERE program, to provide access to state-of-the-art exposure assessment technologies and data resources. There's wanted to give a couple of accomplishments from the hub advancements. So the ChemBioanalytics Science Hub has launched their Exposome Research Coordination Survey. More than 165 labs have participated in the survey to define what they would consider to be scalable analytic and computational infrastructure approaches for doing exposomics in a harmonized way so that people trust what other labs are doing, but also so that things like meta-analyses can be better performed, right? There's a geospatial sciences hub because, again, in addition to, exposomics is the full breadth of exposures, right? And so while some of this work is very much, looking at LC and GC-MS approaches to plasma or sera, right? A lot of this is also looking at structural factors around, neighborhoods and these things, as well as, weather and climate and, wildland forest fires and things like that, which we'll talk about in a couple of slides. And so, I think that the productivity of this network has been really fantastic. And I think it's positioning NIEHS to be able to fund research moving forward that will be, really the top of the line, sort of reproducible scientific work in this space. I need to advance. There we go. And just wanted to mention that several of us will be attending and participating in the Global Exposome Summit that's been organized by IHEN. And so again, getting thought leaders together and really working to follow up to the Exposome Moonshot Forum to accelerate collaboration, shared resource development, and to do that both domestically as well as with our partners internationally. Moving on, I'd like to briefly give an update on our HUE program, Health Effects of Extreme Weather. So the strategic framework around this program has been published and has 5 goals. So this is understanding health outcomes of exposures that are associated with extreme weather and weather-related natural disasters, assessing short and long-term health risks across the lifespan and among populations who may be at heightened risk for harm, developing evidence-based interventions to reduce the health impacts and to improve the quality of life for individuals and populations, maintaining an infrastructure to encourage community engagement, research connectivity and data sharing, and providing training and capacity building for researchers in various disciplines, both in the extramural space, where the HUE program funds, training grants like Fs and Ks, as well as in the intramural space, where we have the intramural component of our HUE work that has been really successful in getting individuals who are researchers at other institutes largely to think about how the health effects of extreme weather might sort of intertwine with the work that they're already doing so that they can, continue doing their work, but view it through this important lens, right? And so that's been a really fantastic way to engage more researchers with this, right? Not necessarily trying to build researchers who are extreme weather researchers exclusively, but rather to get everyone who's doing, important biomedical research to consider how their work might be influenced by this, portfolio of exposures. So the scope of the HUE program currently, and based on what has and will continue to be funded, I was looking at air pollution that's influenced by weather or by disasters. Again, this can in particular be things like wild and forest fires, right? Changing incidence of waterborne, vector-borne, and weather-driven infectious diseases, drought, flooding, really fantastic work around, mold that will sort of take root and grow in homes and maybe sort of unknown and under the radar in following, the following floods receding, right? Harmful algal blooms, snowpack loss, and as we said, wildfires. And so this has really been a tremendous program that's, really prioritizing multidisciplinary research as well as global collaboration. And so the NIEHS director is partnered with the Peter Marks, who's leading the Fogarty Center, which, again, is focused on sort of global health research, really, sort of joining those two things together to, protect individuals from extreme weather through exposure research, risk assessment, intervention science, and of course, training and research infrastructure and data sharing. We had a recent HUE Executive Committee retreat earlier in March. I thought that this went extremely well. We had the director or acting director from 8, I believe, different NIH ICs there participating. And some of the key, themes that came out were, strengthening the workforce in this area, supporting that next generation of environmental health scientists who are interested in this space, Data resources, so expanding infrastructure and platforms that can better support integration of environmental exposure data with health data and population data. And then, Make America Healthy Again related topics around wildfires and environmental disasters, other emerging environmental health threats, and one that I'd want to highlight, which is lifespan and life course studies. And so, some of these data sets that have been put together by CAFE, which is the sort of data coordinating lead extramural program site. I think many of those data sets are sort of ready, shovel ready for looking at things on a maybe 1-year timeline. So things that are kind of acute, like survival after an incident MI, right? And so what we really want to see and what came out of this committee retreat was improving those data resources so that we can look at things like, what is the association of these, health and extreme weather factors on the incidence of various longer term chronic diseases, right? Because, if you're trying to look at a cancer that has a 7-year latency period, right? It's going to be a different sort of expanse of data that one needs. And so these are priorities for us moving forward. All right. Moving on, just wanted to give an update of how we're thinking about environmental health disparities under the sort of new NIH portfolio for this. And so this is really representing kind of a shift to solution-oriented approaches. So advancing beyond documenting disparities, more towards, delivery, right? Implementing scalable and evidence-based solutions, prioritizing interventions that can produce measurable health improvements, and really emphasizing, translation, implementation, and public health impact. Again, this all aligns very well with the NIEHS strategic plan. So, things like identifying environmental drivers of disproportionate disease burden, exposures that may be higher in a population than in other groups and may underlie differences in disease incidence or, difference in disease outcomes. Advancing prevention strategies, particularly those informed by mechanistic science and exposomics, supporting ongoing community engaged research and translational implementation, and accelerating that translation into effective real world solutions. And so, this is an area that I think we are continuing to be engaged in and work toward and funding grants in. And I think that, really what we want is not just sort of documenting disparities, but coming up with solutions or coming up with mechanisms that are really underlying those, right? I wanted to also highlight some work that was done both intramurally and then with our extramural team to put together an OTA. So you all recall the East Palestine train derailment, where a great deal of butyl acrylate and vinyl chloride and other things were drained out of an overturned train and ultimately burned. So this presents a complex disaster response scenario because you're dealing with mixtures with multiple exposure routes, dermal and airborne and in the water supply potentially. And so the National Toxicology Program group and our Division of Translational Toxicology worked to put together a framework for rapidly reviewing and summarizing authoritative literature, doing a rapid scoping review to really identify known health outcomes and evidence gaps for chemicals that were part of the spill, to help inform disaster research and to better communicate health information to affected communities. Now, developing this framework took time, and so some of this work has informed the extramural research that's going on in this space, but I think it also really creates a tremendous framework because, hopefully something like this doesn't happen again, but of course we know that these things do occur in the United States and elsewhere, and so being sort of prepared to be maximally responsive is where we want to be. So I wanted to, again, highlight the trained around that health research program. So NIH through NIEHS is funding a 5-year longitudinal health study that is anchored in a permanent community office in East Palestine, Ohio. It's led by investigators at the University of Kentucky, partnering with folks at Pittsburgh and Yale. And it really serves as a hub for participant enrollment, research coordination, and it's honestly just one of the best examples of community engaged research that I've seen. And so I was fortunate enough to go out there for the grand opening. Dr. Hood, you should be in this picture, and I think they did you dirty and cut you out, but Daryl was like right over at the edge of that.
That's quite all right.
On the right, okay. But I think it was one of the more satisfying things I've had the opportunity to be involved with, and seeing this kickoff. And the community was, they have concerns, they have questions, they have distrust, right? And so having this sort of embedded research office that is led by the extramural community and not the federal government, I think is a really important decision strategically that I think will drive much better research engagement and ultimately, hopefully, improve health and outcomes for these folks. And again, it's looking very broadly, because we don't sort of know what, if any, health effects there will be. But that things need to focus on, the liver and reproductive health and children, but also the elderly and mental health, and stress responses, and so it's sort of broad in that way, but I think intentionally so, and is going to be really an important investment for us to support this work. Also wanted to provide some brief highlights on our Common Fund work. So just as a sort of introduction, the NIH Common Fund makes very strategic investments in time-limited and goal-driven programs that can ultimately really change the trajectory of biomedical research, advancing areas of both biomedical and behavioral research that are important to the missions of multiple institutes and centers within NIH, as well as to the extramural community. And so, this is really an approach to drive innovation and spur advances that might not be possible without a strategic investment, something that's perhaps a little broader than what an individual IC might be able to do on its own. If you may or may not have heard, but our RNomics program has been approved for Common Fund funding. So this is a partnership between NIEHS and NHGRI, and also thanks to fantastic leadership from my deputy director, Dr. Trevor Archer, who really spearheaded getting this up and off the ground, and ultimately now getting it approved. So the RNomics program looks to develop really an essential toolkit for characterization and measurement of the RNome. So we're talking about modifications of RNA. And so, fostering a supportive infrastructure for groundbreaking research. You think about, kind of going from genomics and the genomics revolution, right, to all of the work in epigenetics, and really, in the DNA to RNA to protein space, we've not had, this sort of concerted effort on looking at sort of epigenetics and alterations, modifications to RNA. So the RNomics program is seeking to develop essentially, a toolkit for characterizing and studying the human RNome. So this is RNA sequencing technology, computational tools, molecular standards, as well as a coordinating center for benchmarking and standardization. And I think the time is right for this. It follows on a NASEM report a couple of years ago about the RNome. I think it also aligns with advances in technologies for doing direct RNA sequencing and as well as longer read sequencing. And so I think that the technology and the research interest have kind of come together at a important juncture in time that we are going to be standing ready to address. looking to ultimately, develop RNA-based clinical biomarkers, RNA-targeted therapeutics, and to understand what the environmental impacts are on the RNome, right? Because we know a great deal about, certainly not enough, but a great deal about environmental exposures and their influences on, genetic changes and genotoxicity, as well as epigenetic changes, DNA methylation and chromatin changes. But looking at this in the space of RNA modifications will be really a new frontier that we're enthusiastic about. The other one that I wanted to highlight is the complementary program. So this is complementing animal research in experimentation or in experiments. So the objective here is to catalyze the development, standardization, and validation of NAMs, of new approach methodologies that can ultimately transform how we do research. So we want to better model and understand human health and disease outcomes across populations, develop NAMs that provide insight into specific biologic processes or disease states, validate our mature NAMs to support regulatory use, and I'll mention that a little bit more on the next slide. And ultimately complement traditional animal models that can make biomedical research more efficient and ultimately more effective. So this is a partnership between NIEHS and NCATS. So there are several components here. So there's a NAMS data hub and coordinating center that NIEHS will be leading. So this is the central hub for program coordination, data collection, curation, management, and standardization. There will be funded technology development centers as well as NIH Challenge Awards that will provide data into the data hub and coordinating center. And then that group is going to coordinate with the validation and qualification network, which ultimately is working to, take what is a potentially extremely useful NAM model for research and do the work that needs to be done to have it be something that might ultimately be accepted by our regulatory partners at FDA or EPA. And so that's going to be operating through a public-private partnership, the VQN is, which is being managed by the Foundation for NIH. All right, I think I shall move on. So also wanted to highlight, some of the fantastic stuff that's going on here in the intramural program that we are really excited about, and give a couple of shout outs. So we had our NIEHS Awards and Honors ceremony for our Mentee of the Year, or Fellow of the Year, and our Mentor of the Year. And so that was Jennifer Ish, who was our NIEHS Fellow of the Year. She works with Lexi White and is working largely within the sister study, but on breast cancer epidemiology and molecular epidemiology, and Jesse from our neurobehavioral core. And so we just congratulate these two. And it was a really strong slate of nominees. And so we're excited to see, not just fellows who are going to go on to, I think, be future leaders in this field, but to have our intramural program really supporting the mentorship of these folks and to be able to recognize that. So a few more awards and honors from our intramural program. I'll sort of let you just read through those. One that I did want to specifically highlight is Lisa Ryder, who won the American Medical Association's Nathan Davis Award for a physician working in the government space to do really fantastic work. So she works at our NIH Clinical Center and is an NIEHS investigator who's actually one of the handful that are not here with a duty station at NIEHS in Durham. And she's working at the Clinical Center in Bethesda and just doing amazing work around myositis and other sort of autoimmune and musculoskeletal disorders. Also wanted to note that we had the first annual, so I guess it's going to happen again because it was called the first annual, HHS National Conference on Women's Health. So this is sponsored by the Office of Women's Health at HHS. And so I was asked to speak. And so what I decided to do was moderate a panel And so Natalie Shaw from our intramural program joined us, as well as Susan Sumner, who many of you may know, who's at the UNC Kannapolis campus and directed one of our HERE sites for many years. Joined us to talk about, precision nutrition for public health, exposomics, and Natalie's work, which is really exciting, looking at ovulatory and anovulatory cycles in girls during adolescence, and really helped us to sort of frame our section of this conference on the idea that women's health begins with girls' health, right? And so, and we were fortunate enough to have the second lady come and introduce our session. And so I thought that was really wonderful. We had initially been slated for Friday and were actually moved to Thursday morning because just the enthusiasm around environmental health research was so strong that they really made a wonderful effort that we greatly appreciated to highlight that type of work for this audience. And then, I wanted to take a moment to recognize Bruce Hammock. So I imagine almost all of you know Bruce, really prolific scientist, gifted teacher, and a collaborator who, I think has been really one of the, from what I can tell and hear and all of the stories that I get told, was just really a marquee collaborator and made collaboration easy and fun and rewarding for everyone that he partnered with. And so Bruce passed away, and we wanted to take a moment and celebrate him and his life. He did also direct our first NIEHS Superfund basic research program during his time at UC Davis. And so just take a moment there and remember Bruce. Okay, we're going to move to questions here shortly. I think I'm actually doing pretty well keeping time. I'm proud of myself. So the NIH-wide strategic plan for the next period for fiscal years 2027 to 2031 is in development. So NIH is in the process of developing this strategic plan. They'll be seeking input from the public. Actually, I believe that the notice and portal for this may have gone live yesterday. We can follow up with information on that. But the leadership at NIH is going to be introducing the process for developing this next NIH-wide strategic plan, outlining, a sort of high-level framework and answering questions. So there's a meeting scheduled for this on April 8th. So I just wanted to let everybody know that this is happening and really encourage you to participate because, it sets the stage, right? It sets the stage for what we're going to be looking at and looking to do moving forward on a, NIH-wide scale. All right, so we're going to go ahead and do some questions. We don't have to do these specific ones, but if nobody has questions, which I suspect is probably not the case, I have some ideas of things to get the topics going. But things that I'm interested in, as the director here for the last 5 months, are hearing about some emerging research areas or technologies that you think will move environmental health sciences forward in the coming years. How you see our mission and strategic plan advancing the goal of identifying underlying causes of chronic diseases, work that you or your peers are doing, and how you see that fitting into these strategies and these goals, and thinking, critically about technology or approaches that you've seen in other fields, and how that might be able to be incorporated into environmental health research, because I think there's a lot of great work going on out there, and I think the idea of being able to bring in some of those toolkits or approaches would be really beneficial for us. And then ultimately, just barriers in terms of, your work, the work of our, next generation. We're working obviously to get grants out the door here we have some updates on that, and I'm going to leave that area for David to talk about. So I'm going to turn off my screen share so that I can see folks' faces. If you want to start raising hands, we can call on folks for questions, and I think we have a little more than 10 minutes. Daryl, Dr. Hood.
Hi, yes, thank you very much. I really enjoyed that recitation. Relative to the first question that you have on your sheet, and I'm really, really happy that you brought to the forefront this concern that you have as it relates to population stratification in, for an example, longitudinal epidemiological studies. That brings to the forefront this question, and we have to reconcile how we're going to deal with that. And hopefully you all can do that when you go over to the Exposome Global get together. And what I'm speaking of is this underrepresentation of input that's going into these large language learning models, and it's being magnified and amplified such that, as you might imagine, that will have a devastating impact on, personalized medicine. I mean, I can go down the list. Yeah. And so that being the case, how do you see the Institute contributing to reconciling that? That's something that we're going to have to do before it gets out of control, if you know what I mean.
Yeah.
We can't. Yeah. So that's my question.
No, I appreciate you raising that important point, and I think it's something that we are definitely thinking about. I think there's probably multiple different approaches that you could take, but as an example, right? If you look at the All of Us study, right? The All of Us study was very intentionally designed to be not only sort of population representative in terms of geography and other things, but to enrich for racial and ethnic minorities, and to try, the best you can with participation rates being what they are, to recruit individuals from lower socioeconomic brackets, right, to try to get this sort of full representation. And so, I think from the extramural space, in terms of grants that we would review and potentially fund, obviously we want to see those grants, including, patient sets and biospecimen samples that are going to lead to their results having external validity, right? And that will lead to their results being, not just useful in isolation for, one specific question and one specific group of people. And so I think that that's one thing we can make a priority. But the other is, I think layering some of the exposomics and metabolomics work on top of All of Us, it would be a strength, because they've already done the legwork, right, to design that group as being sort of representative and being able to address these issues around stratification and ancestry and socioeconomics and all these factors. And so as we work toward funding more exposomic metabolomic type work, I think being able to build that on top of All of Us when the opportunity might arise is one approach that we're enthusiastic about, because also there, then that data becomes part of this public repository for other researchers to access and engage with. And I think hopefully that, through a sort of litany of approaches, but keeping that at the forefront of our minds, that we can prevent that sort of downstream problems that we could sort of rapidly get to, as you highlight. So I thank you for that.
Sure. Thanks.
So thinking about, the mission and the strategic plan and advancing, the goals of NIEHS, which, both I think largely remain the same, right, as they have been for years, but have now kind of, I believe, been placed front and center, right, is really an opportunity for us. And so, I see we have a hand up. I can't actually see who it is.
Steph Cormier.
Oh, there we go. Steph Cormier. Yes, thank you.
How you doing? So I actually love the things that you laid out in your talk. One of my thoughts is the life course studies. As I mentioned, I'm interested in infancy and long-term health, respiratory health impacts, actually. But one of the big problems is actually grants don't actually cover lifespan approaches. And so I don't know if there's going to be a method or something set up to where we can actually start incorporating longer term studies from NIH. But I mean, that's the biggest problem, right? You have to piecemeal grants to do a life course study.
And of
in
course,
the US.
you know, you have this also, I mean, we're kind of past this now, but like in that cohort construction space, right? Not a lot of new cohorts getting launched in the last 15 to 20 years, right? But even when they are, you know, you have this problem of, well, you know, you do your competing renewal, and it does all right, but you got to go back in with your A1, and maybe that does well and it gets funded, and maybe not. And when you have those breaks, right, in funding, and you have to potentially lay off staff, right, you're not able to continue that sort of, you know, rigorous contact with folks. And we know that when we start to lose contact with people, you know, for more than 6 months, you just, you never get them back, right? And so I'm very sensitive to this problem. I think that, you know, existing ongoing cohorts are things that there's huge value to, and we will be making concerted efforts to really try to support as best we can. I also think that there are intramural NIH-wide cohorts that are being stood up that are collecting enough types of data, right, that grants that leverage these are likely to compete very well. Things like, you know, layering something on top of ABCD, right, or the ECHO program, right?
Right.
Exactly. And so I think that you know, for anyone who has a biobank and has a cohort, knows that they should be sharing specimens for research, right? But you have to prioritize what you share. And I think we're at a good point now because, you know, I never wanted to give away half the DNA from an individual we enrolled so that somebody could genotype, you know, 2 SNPs, right? 2 markers, right?
Yeah.
But now that we're in the exposomics, metabolomics, RNomics space, you know, that sort of decision tree gets much simpler. And it's, if you're going to do something high throughput, broad scale, and we can have you be sharing those data back to these sort of centralized resources, then it's time to move the needle on distributing those specimens and facilitating that type of research. And so I think, again, we're just at a nice time point there where we need to sort of change our mindsets around these cohorts and how we share samples to say, well, you know, like if the science is good, but also if the, you know, if the readout is broad enough, like what do you need to save these biospecimens for if you're generating all the data? Now, obviously some new things come up and, you know, there is that, but very, very appreciative of that comment. So.
Thank you.
Maria, did you have a question? Yes.
I do. I so support the focus on actionable health solutions. And just wondered, you noted that the focus when we're looking at environmental health disparities, we'd be focusing more on approaches to solutions versus documenting disparities. And I just wondered, since understanding, you know, the disparities through documentation and data is a key to the solutions. What does that mean?
How do you disentangle that? Yeah, I think, you know, in terms of, I think ultimately what we want is to, you know, deliver interventions or, you know, approaches to reduce risks and, you know, and move that needle for people. I fully acknowledge, and I think our framework for doing this acknowledges that understanding etiology is an important part of that, right? And so, you know, what we want are things that, you know, get to sort of etiology at a mechanistic level, right? And so if you can sort of really dig in on, you know, mechanistically, how is this working, so that we don't feel like, well, you know, it's, you know, something is driven by SES. It's like, well, lots of health factors are driven by SES. that's a pretty hard intervention point. There are things that people are doing to intervene sort of at that scale. That's more an NBER thing and sort of outside the NIEHS space. But, when you think about environmental exposures that have a different distribution in the population, I think, like a great example is intramural and extramural work done in like chemicals and hair straighteners, right? So these are utilized at different frequencies among different populations based on, race and ethnicity and cultural factors and all of these things. And then when you see that there is also a difference in the incidence or in the outcomes of a specific disease, or even the incidence of certain molecular subtypes of, a disease, really trying to get those pieces connected to understand like what those mechanisms are, and then, strategize on that of where the intervention points may be. And, this can, ultimately, be anything from, oh, well, like we need to, have some small, this is affecting things in this way, and so this small molecule inhibitor might work. Like that's certainly one area. It could just be, these things are, hazards to health, and so this is the key population that needs to be, educated about that, where we need to intervene, right? And so what does that messaging look like, and how do we do that in a sort of responsive and community engaged way, right? So that it's not just, shouting into the void and no one hears you, right? Because, you can find really important stuff and learn really important things. And if no one's listening, no one is helped. And we are very sensitive to that. And I think that's one, but not certainly the only reason for why we like to see our community engagement work not just be at the time of discovery and delivery of information, but in the planning, right? And what are the things that are important to these communities to know about and understand, and have that work really engaged throughout, right? Community engagement front to finish. And then again, that's why I was so thrilled with the work that was being done in East Palestine, because they had folks involved from the very start. And I went out there for 2 days, and we did a ribbon cutting. That was the last thing, but the much more interesting and honestly enjoyable thing was doing all the listening sessions, right? Going to a high school auditorium and listening to several hundred members of the town. It's a small town of 4,500 people maybe, tell you what their concerns were, right? And the folks from Kentucky are frantically taking notes, and then afterwards there's we debrief about these, and really a great, I think, approach, and we'd like to see that as a model, right, for other work.
Great, appreciate it. Thanks.
Yes, absolutely. I think we'll have time for one more. Yeah.
Yeah, I think we can do one more. Koken?
Hi, Dr. Walsh.
Hello.
Hi. Thank you for a nice talk. I appreciate the points that you made. I just have a comment, another question. As a practicing physician and a pulmonologist, we obviously have we have to deal with a lot of lung diseases that are caused by the environment. Some of the exposures we know and others we don't. I think exposome studies will enable us to learn more about it and probably uncover some of the exposures that we never thought of. But at the patient-physician interface, we really don't have time to explore, the environmental exposures, both on the occupational side as well as the, non-occupational side. So I think this also needs to be at some point addressed about the help we can provide to these patients. All the research we do is great, but how do we make an impact on the individual patients if we are giving less and less time to spend with the patients?
Yeah, I appreciate that point. I mean, I don't know how long your appointments are, but I have the terrible memory of, so my wife's a pediatric dermatologist, and does a lot of atopic dermatitis, eczema work, and also patch testing. So occupational exposures, patch testing for immune reactions. So those appointments are, I believe, 45-minute appointments, but the rest of the week she's running 5 12-minute sessions a day, or 5 12-minute sessions an hour, right? So every hour you're seeing 5 patients for 12 minutes. And you're right, that doesn't leave time to answer all the medical questions, much less to incorporate new research and environmental health recommendations and all of these pieces. And so you're right. And I think that is a structural issue. And I will make a note, David, I think it's a thing we talk about, with regard to our K-12s maybe, because that's a really solid group of folks invested and interested in this topic, and could be a great resource for young folks with ideas in this arena, right? So thank you, Kogan. Yeah.
And I think we'll go ahead and take Carrie's.
Okay.
Thank you so much. As a civil engineer, Dr. Walsh, I'm really well aware of the impact of the built environment on health. From thinking about the community's investment in sidewalks and parks and access to exercise in nature, to the East Palestine railway disaster, to the need to retrofit our public buildings, including schools, to make them a more healthy environment, particularly thinking about the effect of older and modern building materials. What is the opportunity moving forward to link together NIH and NIEHS research with the built environment and setting goals for a better design that results in better public health?
Yeah, I think this, really gets to both an opportunity and a challenge of what we've, lovingly now referring to as exposomics, right? Because, it's so multi-layered, right? Exposomics is, your individual exposures. And so that's both like things you're actively putting in your mouth, is your diet, right? It's also, things that are coming to you from the water or the air or whatever that may be, as well as sort of these, structural things. And so some of those I think are going to be measurable. I think like mold in homes, or, how good your air filtration systems are, and, how that impacts individuals differently if they live, near or not near, extreme weather events and forest fires and things like that. I think that we really are looking to encourage multi-layered data integration, platforms for doing that, new approaches to try to look at these things. Obviously, like there's been a great deal of work over the years sort of incorporating exposure and genetics, right, in the G by E space. But I think, it's sort of an E by E space of individual level exposures and community level exposures, and working up from there. When you did ask about partnerships with NIH in this framework as well, I would note that I had mentioned like the ECHO group, right? They are collecting all of these types of things, fantastic data collection, not just acquiring cord blood serum and newborn heel sticks and maybe breast milk from mom, but also asking, validated surveys about bullying, right? And in schools, right? When these children are older. And so I think, we really want to be very cognizant of the opportunities that are there with those existing cohorts, as well as, I think there are actually more opportunities for partnerships with NIMH than perhaps we realize or have realized. And so part of that is, NIDA and NIMH, and, NIDA's leading ABCD and HBCD in these cohorts. I think about, again, something that someone mentioned to me in East Palestine, which was that they used to hear the train all the time, and it was something that they blocked out. Like, you just can block out a noise and it becomes part of the background. And now, like they don't block it out anymore. And when they hear the train, they like tense up and they right. And that was, I don't know, sort of eye-opening for me to sort of think about. And, again, I don't know how you exactly measure some of these things, but they're smart folks, and I encourage them to figure out how you do that. But, that's just sort of one example of some of the aspects that I think you might be getting at, that we are open to understanding, exploring, and, that potentially, are areas for intervention.
Thank you.
Thanks, Carrie.
And with that,
David.
I think we probably should move on to my presentation, just so we don't get any farther behind with our agenda. And before I get started, Ashley pointed out that we should state publicly and for the record, clarifying roles. Welcoming Kyle as NIEHS director. In that role, he is the designated chair for the National Advisory Environmental Health Sciences Council. And I will continue in my role as the director of extramural research here at NIEHS as the executive secretary of council, and I am the designated federal official for council, just clarifying that for the record. It's my pleasure to introduce or to present an update on the extramural activities since our last council meeting, and see if my slides will advance. There we go. So I'm going to cover 5 broad topics today. Fairly quick staffing updates, just some comings and goings in the division. We will be taking a vote on the second section, which is the council standard operating procedure and council delegated authorities. That's a required annual vote that we take. So if you want to go ahead and get logged into your electronic council book, we'll be there before you know it. I'm going to present a wrap up of our fiscal 25 grants budget. And some details on that from the NIEHS side, and then also have some discussion about our fiscal 26 funding strategies, our approach to managing the grants budget for this year. We'll go hopefully fairly quickly through some policy updates and some news to note from the NIH side, just to make sure that the community is aware of how to access information and some of the changes that have recently been promulgated. And then we'll close up with some highlights from DERT, some things that we've been working on since the September council meeting. So getting into our staffing updates, we had one individual who left DERT, Caleb Rogers, who was one of our grants management specialists. He resigned during the furlough to take advantage of some other career opportunities outside of the government. We miss Caleb, and we definitely miss being down a grant specialist, trying to process the awards and getting those out. We also, just last week, Kendra Morrison, who was a member of our scientific review branch, accepted a reassignment to go back to the Center for Scientific Review and resume her duties as a grants technical assistant, supporting the reviews for our research projects. So we thank Kendra. They are in desperate need of help up at CSR, and so we really appreciate her willingness to be reassigned. We do have one new staff member in DERT, and that's Lisa Marie Adiaba. She joined us from the former Environmental Health Perspectives team in December. With a reassignment internally. She is working with Ashley as a program analyst, doing a lot of the duties that Nicole Popovich used to do, supporting the division leadership committee and also coordinating some internal data calls. She's our highlighted topic coordinator and is playing a role in publication of our grant notices and those kinds of things. So she's kind of a jack of all trades, helping out with coordination of activities within the division. And she probably will be interfacing with council on some activities going forward. The second bit of business is to present the council standard operating procedure and the council delegated authorities. Again, we will be taking a vote during this section. Just a reminder of what this is. So we are required to have a formal documentation of our advisory council procedures. So we developed last year a standard operating procedure that codifies the way council operates as a routine set of business. Included within that is the annual approval of the council delegated authorities that are normally responsibilities of council that we ask for you to give DERT staff the ability to process those. So we have made a number of changes to the council SOP this year. So the council members have that available to them in the electronic council book, both as a clean copy and as a tracked changes version that highlights all of the edits that we have made. Most of the edits are fairly minor, clarifying some existing procedures and aligning with the structural changes within NIH, no longer having a dedicated review branch within DERT, reliance on the Center for Scientific Review, some changes to committee management and those kinds of things. The more substantive changes to the council SOP include the addition of processes and procedures around the new category of grants, which are the competitive but not discussed applications. We will have Bruce Reed from Center for Scientific Review joining us after the break, and I'm sure that this will be one of the topics that he addresses there. Those applications are being shared with council in their own block within the on-block review categories, so that those will be approved by council, reviewed and approved by council for the second level. We're also clarifying in the council SOP that the truly not discussed applications, those that are identified as not being competitive for funding, are not routinely presented to or discussed with council in closed session. The cases where we will bring those to council are cases where there's an appeal of those actions, or if and this has only happened one time in my 25 years at NIH. There is an application identified as streamlined, not discussed, that we are actually proposing consideration for funding. That's a very rare circumstance, but it does require council review. We have also made a little bit of a streamline to the SOP to be in alignment with NIH policy. We formerly required a vote from council on applications that are identified as concerns. Those concerns include things like human subjects, women, and sex and composition, children's research, any vertebrate animals research concerns, or budgetary concerns. NIH policy actually says that those applications need to be brought to the attention of council, but it does not require a formal vote from council for us to be able to resolve those concerns. So that'll be a minor change to the operation in the closed session for that. And then the most, to us, significant change is a mindset change around, we had a former process that allowed us to bring certain applications that are time sensitive to an early council concurrence. That early council concurrence was fairly prescriptive in what we could bring to early concurrence, and was mandated to use the electronic council book for voting, and was only done before the council meeting. And on discussion with other institutes about their practices around obtaining council concurrence on applications that had some time sensitivity, as we are dealing with the delays that we've had in peer review and summary statement availability and those challenges, we learned that a couple institutes use what they refer to as an interim council concurrence, which allows them much more flexibility to bring applications that don't require special council review and formal discussion during a council meeting to council's attention to obtain concurrence with the initial review group between meetings. So the procedure for this will be an email vote rather than a vote during the electronic council book. And we will annotate the consideration of those applications in the closed session minutes for whichever is the most adjacent council meeting. So that could be a revision of the prior council minutes or inclusion within the next council meeting. And this will give us much more flexibility to manage the timing of awards between council meetings. Then finally, we are required annually to get council's approval of our council delegated staff actions. We are not proposing any changes to the council delegated authorities. Again, these are normally council responsibility that are relatively straightforward changes to existing funded, already approved applications. This includes a change of institution, so the movement of a grant from one grantee institution to a different grantee institution. Typically, that's associated with the movement of the PI. The second is the change of a PI. So if a principal investigator retires or unfortunately passes, or for other reason needs to permanently leave the project, we ask council to give us the ability to approve the new PI. A continuation of a grant with an interim PI. This is typically either due to a PI going on sabbatical or having a long-term health issue that does not require a permanent change of the PI. And then two processes for extending grants, either without funds as a no-cost extension, or an extension potentially with funds, what we call a with-cost extension of that grant process. So those are council authorities that we ask to be delegated to NIEHS staff. With that, I'm going to pause. I'm going to see if there is any council discussion of the SOP or the CDAs. So if so, please raise your hand, and I'm glad to have that discussion. If there is no discussion, if I can please request a motion to approve the SOP and the CDAs.
Motion to approve.
Second.
Thank you, Steph. And a second from Daryl and Carrie. And if council members can please vote, we'll try to do this in the ECB.
Yep. So you should be able to go to the council agenda for use in meeting and scroll down and click on the NIEHS Council SOP and CDAs and click yes or no. And I can see the votes this time. I don't see any in yet, but let us know if you have any challenges.
It's working.
Yay.
All right. Mm-hmm.
It's not working for me. I see the yes or no, but I can't click on them.
It worked for me. Mm-hmm.
We have a quorum. Okay. And then I guess, Dr. Matlu, did you want to register a no or a yes or an abstention? A yes.
Yeah, yes.
Okay.
All right. Thank you, Gokan, and thank you, council. So then I'll move on and present our fiscal 25 budget wrap up and our approach to managing fiscal 26. So this is some truth in advertising. Last year, NIEHS was assigned 1,330 applications which were reviewed. That led to the issuance of 225 competing awards. So that 225 competing awards is slightly lower. So we received 110 fewer applications. And let me use my pointer since I'm kind of jumping around here a little bit. So we received 110 fewer applications than we did in fiscal 24, and we issued 60 fewer awards than we did in 2024. with that budget, we managed to maintain actually a fairly positive success rate, 12.3% success rate for our R01s, 12.9 for RPGs. It was slightly lower than fiscal 24. So in 24, our R01 success rate was 13.8%. I'll present some more data on this later, but we did issue 57 awards as multi-year funded. That was a total of $58 million that we invested in multi-year funded awards. That is a significant uptick from prior years. But we did, while we were still operating under the concept of a pay line, we did have an effective pay line last year of 11%. We funded all applications except for a couple things that had already been funded up to 11th percentile. And for the first year, we formally had a higher pay line for early stage investigators. We had a 16%. We funded all USI applications up to 16th percentile for our early stage investigators. Looking at the distribution of those awards between the different mechanisms. First, our total award budget, this includes both competing and non-competing awards, was about $466 million. That was about $20 million more than we had funded previously. And as Kyle shared, with our budget having been flat, this is actually a pretty big, significant increase in the amount of grant funding that we were able to issue in fiscal 25. Our distribution rate remained fairly consistent relative to previous years. Our RPG budget actually is a slightly higher percentage of the budget overall than where we've been recently, $326 million in RPGs relative to 284 in fiscal 24. Our other budget is now our second largest budget line at $56 million, up from 44. This does include our health effects of extreme weather, which was a significant investment in the other research line last year. Our center's budget decreased significantly because we did not have competing Hughes centers last year. So we went from $73 million in our second largest budget line in centers down to $43 million last year. Our SBIR is a congressional mandate at just under 5% of our budget, and so that was flat at $23 million. And our training budget, our NRSA budget, ticked down just a little bit to $19 million from $21 million in fiscal 24. Breaking down our RPG budget, so these are the actual research awards supported by the NIEHS. Again, this has been fairly consistent. About 75% of our RPG budget in the R01 line is pretty consistent. R21s, R35s going down, U01s down to just one or two UH2s and UM1s that we supported. We did, last year was the first year ever that we've supported our 50 awards. Those are kind of a staff scientist research award, so an independent award for non-faculty level researchers that we supported through an ODSS program last year. Breaking down our competing versus non-competing awards in the research project grants. We issued 125 competing RPG awards at a total of just under $100 million. 12 of those 125 were solicited research. And as you can see from a budgetary standpoint, our non-competing awards represent a much more significant portion of our budget, a total of 421 awards at $227 million, and 103 of those non-competing awards were solicited. So we're continuing to see a shift in our portfolio from solicited applications into investigator-initiated, which we think is going to continue significantly going forward. As I said, multi-year funding was a rather significant uptick in fiscal 25 with the guidance from OMB that we should increase multi-year funding to decrease those non-competing commitments in future years. So last year, overall, we did a multi-year funding of 69 total awards. That included a few non-competing awards, 7 non-competing awards that we multi-year funded for a total of $71.6 million from our HHS appropriations. Just for reference, in fiscal 24, we issued a total of 11 multi-year funded awards at a total of $4.5 million. So more than a tenfold increase in the funds going into multi-year funding. By comparison, our incremental awards, we issued 482 incremental awards at $254 million, only 68 competing awards, and 414 non-competing awards as incrementally funded applications. Our solicitations, we only had 5 solicitations that we funded awards from last year, 3 NIEHS programs, really only 2. The Victor, the final year of the Victor awards was last year. We issued 3 awards for $2.2 million. And we had 2 different solicitations cycles for our time-sensitive research opportunities. So a total of 6 awards issued through that program for about $2.5 million. And then we also participated in 2 of the ODSS, issued awards for 2 ODSS programs last year. The Sustainable Software, 1 award, and the 2 R50 Research Software Engineer Awards that I mentioned earlier. Looking at our interior appropriations, our Superfund appropriation, and this was fairly consistent with previous years. We issued a total of $76.1 million, or had a $76.1 million budget for interior programs split between the worker training program and the Superfund research program. So all of the worker training programs, the U45s, the UH4s, which are not included on this slide, the pass-through from the Department of Energy, and their small business program competed last year, a total award of $26.6 million. And then we had 1 competitive cycle of the Superfund P42 awards and 1 non-competing cycle. We did multi-year fund a couple P42s to prepare for the potential 40% budget reduction that we have. That was the president's budget and the House mark last year. We multi-year funded some P42s as well. So the total SRP investment was $49.5 million last year. Changing gears to talk about how we're going to approach fiscal 26. I wanted to start by acknowledging the news that has been out. There have been a couple articles in Science and some other venues about the significant delays in the issuance of awards from NIH. I think last update I saw only 1,200 competing awards had been issued across all of NIH as of last week. And I just want to acknowledge the reality. So as I've presented in previous years, we've put significant effort into trying to balance our workload throughout the fiscal year with a goal, which is never going to be completely attainable, of doing a quarter of our awards for each fiscal quarter of the year. And as you can see in 23 and 24, we did a pretty good job of balancing it with Q3 being our largest number of awards being released. This is both competing, non-competing, and prior approval awards. So non-monetary awards are captured here. Last year, we actually started out at a very strong pace, best we'd ever done. We maintained a little bit of momentum early in the second quarter before everything stopped. And then as we got the approval to issue awards, to do the negotiations of projects for alignment with administration priorities, we were able to increase the number of awards in Q3, and we were successful in Q4 of getting all of our awards out and obligating all of our funds to our awards. Our total number of awards, historically, we've been around 1,100 awards issued per year. Last year, we were at 969, so slightly decreased number of total awards issued. And as you can see this year, we have, this is a couple weeks old data. We have released more awards since then. Under 200 awards issued, or just over 200 awards issued when I generated this slide. So why the delays? So some of it are things you're aware of. The shutdown of the government in October and the first half of November delayed us for 6 weeks in issuing awards. The delay in holding this meeting, normally this would have been scheduled a month ago, and we would already be in the process of issuing competing awards from our February council round, which clearly we are not doing yet. One thing you may not be aware of is that to make this success of getting all of the awards out, we did not process any non-monetary approvals in Q4 of last year. So we started the year with placing a priority on finishing up those fiscal 25 non-monetary approvals, the no-cost extensions, the change of PIs, those kinds of things. So we had about 2 months worth of work to do when we started the year, then started the year with a month and a half delay in being able to do anything. We are still somewhat feeling some pinch of the changing HHS and NIH policies and priorities, the need to negotiate applications to be in alignment, and those sorts of things are slowing down the issuance of awards. And then finally, the OMB and the Department of Treasury have implemented a new procedure this year for getting the fiscal 26 appropriations to federal agencies that both delayed getting funds for a month, but also going forward is going to make life significantly easier for us. So we did not receive a prorated apportionment of funds, which would have allowed us to increase the release of awards following the approval of the budget in January. We have this week, however, received our full year appropriations. So yesterday we released, I think, 17 awards with the word that we are getting all of our money for the remainder of fiscal 26 this month. So we have queued up a number of awards, I think between 50 and 60 awards that are ready to go as soon as we receive notification that the money is in the bank for us to support those awards. I do want to also have some truth in advertising that when we click release on an award, it goes through a number of additional approvals and notifications of Congress and what have you of those awards. So it can be several days to even a couple weeks after we release the award before you actually receive the notice of the grant award. How we are approaching making our funding decisions for fiscal 26. We are following the NIH Unified Funding Strategy that Dr. Bhattacharya presented when he visited Council in September, and there's a recent piece that was published by several of the Institute directors, QR code here that you can scan, that describes the Unified Funding Strategy. Which is the TLDR version of that is that we no longer use the concept of a firm pay line, that peer review and the numeric score of the application are an important factor in funding decision making, but they are not the only factor that is considered in funding decisions. Instead, we're looking at things like alignment with the NIH and NIEHS mission, some priorities around early stage and new investigators, programmatic balance, identifying gaps in our portfolio, geographic balance, and then mandated stewardship and appropriate oversight of our portfolio. So that's a lot of words to say. Honestly, at NIEHS, it's not a very significant departure in our approach to making funding decisions. We have always looked beyond just the numeric score in setting our determinations of what we fund and what we don't fund. As we've discussed in the past, we've always had a reserve of our budget for what we formerly referred to as raise to pay applications. And did relatively few skips within our pay line. Again, we no longer have a defined pay line, but I think that you will see the funding decisions that we are making will be very consistent with what we have done in the past. And then from a policy standpoint, some of our procedures to manage our budget this year, we are no longer implementing our standard set 15% administrative reductions across the board to all competing awards. That was a budgetary gimmick that created more problems than it solved. We will be looking at the individual application and the budget request, and we will be applying programmatic cuts as we think that there are costs that are not necessary, but we will no longer be doing a standard across the board cut. Our non-competing awards will typically be awarded at committed levels. We are not going to be doing a standard across the board reduction to our non-competing awards. We did for the first cycle, our February 1 starts for the P42s, we did implement an across-the-board cut just because of the potential for a 40% budget reduction to that program. Once we got word that 40% budget reduction was not going to be implemented, we have revised all of those awards back up to committed levels. And then finally, I just want to acknowledge that our multi-year funding strategy is still somewhat under development pending final guidance from the NIH Budget Office on our procedures and targets for multi-year funding. We know we will not be doing more than what we did in fiscal 25. It's unclear whether we will be held to our fiscal 25 numbers or not. And then finally, I just want to reiterate, this is not change in any of our strategies, but continuing to emphasize our efforts to manage unobligated balances in our portfolio. They are significantly reduced from where they had been historically, but we are continuing our processes of any awards that we have as a non-competing continuation that have more than 100% of their annual budget as an unobligated balance. will be receiving a mid-course no-cost extension. Any awards that have between 70 and 100% of their annual budget as an unobligated balance will undergo either a restructuring where that unobligated balance is allocated across the budget. So we will receive a reduced award this year, but increased awards in the future. So the net balance is unchanged, or in particularly egregious cases, that award may be offset and the overall award for that project will be reduced. And then also implementing our procedure of, this is the second year of a competing renewal. So that's a type 2 application. We will be offsetting the unobligated balance from the approved and accepted financial report from the previous competitive segment. And I've discussed all of this in much more detail with figures and numbers for scenarios in the September Council. So if you want to go back and look at those slides as a reminder of how all of that actually works. Next, I want to go in and do some policy updates and some news to note. Hopefully we'll go through this fairly quickly. It is a little bit convoluted. So there is a significant effort underway to centralize all extramural information on the grants.nih.gov website. So the historic information that we've had on the NIEHS webpage around our grants programs, our approaches to different aspects of our portfolio, all of that is being centralized in a formulaic website on the grants.nih.gov website. So there's a screenshot of this here. This site is still under development. It is continually being updated and enhanced as they are going through the process of the centralization. But this site is really your one-stop shop for all information relating to grants, not only at NIEHS, but across all of NIH. Associated with this, it's actually, ultimately, it will be easier, but right now it's a little bit of a challenge to find research opportunities. So you do still have this website here. That will take you to the grants.gov information on all NOFOs that are released. If you click on funding, there's a drop down that's find a fit for your research. You'll see here two separate web pages or links. The first is to highlighted topics. Much of the substance of what you have formerly seen from NIH in RFAs is now going to be done through highlighted topics. And I'll talk a little bit about those. I've mentioned them in previous council meetings, and I'll talk a little bit about them in a couple slides. There's also on this site a link to Institute Center and Office Profile. So if you click on that and scroll down to NIEHS, you'll see, so here's the QR codes to get to those sites. So this is the NOFO listing. If you click this button, you'll see this side. And if you click on Institute Center and Office Profiles, scroll down to NIEHS and click on the link there, you'll see this web page, which is the new NIEHS grants web page. This information is standard across all institutes, so all institutes have a very similar page and very similar content on this site. It has our mission statement pulled from the strategic plan. It has general topics of interest. This is, I think, is actually a very positive thing. This is a cut down version of the floor of the research that NIEHS will support through our grants programs. We have a formal document negotiated with other institutes and the Center for Scientific Review called our referral guidelines. That is not a public document, but it sets the foundation of what is NIEHS research and how does that differ from NCI, NHLBI, NIEID, any other institute. So this general topics is a pared down version of what those referral guidelines are. And it does include a description of the research that is supported by the Superfund Research Program and the Worker Training Program. This site also includes a link to all of the funding opportunities, these funding opportunities that NIEHS participates in. It includes any highlighted topics that we participate in on there. And then importantly, this link for additional information by funding category. This is where you will find the information for our interests on every category of funding that is solicited through a parent announcement or a program announcement. So if you're interested in what does NIEHS support in conference grants, in SBIR applications, in administrative supplements, this is where you will find that information. That is being filled out right now. Right now, I think the only ones that are live are administrative supplements and SBIR, but we are every other week, we have a deadline to provide more information on other budget mechanisms. So that will be populated in the not distant future. And that is critical information for you to understand what NIEHS is interested in doing and is not going to be doing in those different budget lines. So there's QR codes here for those sites as well. Finally, the last thing I want to include here is important information about where you find notification about policy changes. They are coming still fairly fast and furious. This is still being captured in the NIH Guide for Grants and Contracts. NOFOs are no longer being captured in the NIH Guide for Grants and Contracts. But the weekly email that you can sign up for there will include all policy updates that are coming out from NIH. And again, that's available on the grants.nih.gov website. As you can see, there are many of them that have been released since our September council meeting. I just want to highlight a few of them, a total of 9 of them that are ones I think that are most relevant to our grantee community. The first, most recent, is the updates to the data management and sharing plan. This replaces our old data management sharing plan guidance with a new form that is primarily yes, no, and short answer questions, standard across all grant mechanisms. An announcement, so there's, I'll show on the next slide, implementation of new common forms for publications, bio sketches, what have you. The implementation for those has been delayed to the next receipt date, council round. An important notification that if you have prior approval requests, those must be submitted through the prior approval module. If you send an email to your program officer and grant specialist about a prior approval, it will not be processed. You have to submit it through the prior approval module. A reminder, this is not new policy, but it's a reminder of increased scrutiny. Your application, when you click submit on grants.gov, it must be complete and compliant with NIH policy and application instructions with all of the criteria of the NOFO at the time you submit it to grants.gov. If it's not complete, it will be returned without review. there will be less opportunity to correct mistakes post hoc. Part of the effort to reduce burden to the applicant community and to streamline some processes, an announcement that we will no longer require institute a pre-approval of applications requesting over $500,000 direct costs per year, or requirements for letters of intent for programs. We used to require these for our time-sensitive program, for our conference grants. We no longer require those. There's the announcement of the updated bios sketch and other support documentation that has been delayed. And then finally, 3 processes or policies that were announced in September around bio and data repositories, biospecimen collection that I think are important to those communities. And then finally, the guidance was approved or released in September on the new PF5 mechanism for NIH-funded international collaborations. So that PF5 mechanism was announced in September, and the templates for those have been released. Then I'm just going to close up fairly quickly on some DERT programmatic highlights. So we've had a busy several months since September. We've had grantee meetings for several of our programs, including our P30s. Not technically a grantee meeting, but the Superfund Research Program, the TAMU Center hosted a Superfund Research meeting in December. Our collaboration with NCI on the cohorts for environmental exposures and cancer risk held their grantee meeting also in December in Michigan. Kyle presented the East Palestine Train Derailment Study kickoff meeting in East Palestine, Ohio in February. We held the CURE Consortium meeting just a week and a half ago. The NIH Data Repository Knowledge Base, so ODSS program, held a grantee meeting at the beginning of March as well in New York. And then, as Kyle mentioned, in response to Gokin's question, we had a meeting last week of our PRESH alongside the CDC PASHU meeting last week. This is our K-12 program to support research activities for clinicians to work with the PASHU. So they're clinicians within the PASHU working in environmental health research activities. The PASHU scholars are they are the most amazing group of individuals. They're so impressive, and the work that they are doing is so outstanding. I'd love to see an expansion of this program and our involvement with the K-12, and I think it can help with some of Gokin's concerns and things that we've talked about at prior council meetings about how do we better integrate environmental health into the relationship between a clinician and their patient. The worker training program has actually held several meetings since September, so they get their own slide. They have held 2 grantee meetings and other engagements. Then they've got some meetings coming up in May and October as well. They've also, the worker training program has released a new video on their disaster training activities. So there's a QR code that you can access that information as well if you are interested. Our Superfund program, so Kyle highlighted the complementary Common Fund program. Our Superfund program in their Risky Learning webinar series did a deep dive into NAMS, highlighting the complementary program as well as some SRP-related activities in both in vitro screening systems, NAMS, as well as some computational approaches to advance toxicity testing and screening. And finally, close out with an announcement that one of my babies, the Human Health Exposure Analysis Resource, held its final steering committee meeting in December of last year, bringing to an end 10 years of support for our infrastructure program to encourage exposomics research through both the CHEER and the HERE consortium. And I just want to highlight some of the real accomplishments. So a big piece of this has been the support for projects. So throughout the 10-year period, CHEER and HERE supported 55 investigator-initiated studies. So that's additional analyses to NIEHS, NIH-funded projects. But we also formed partnerships with other NIH efforts. So we did 28 studies for the ECHO program. We've done 4 projects in support of the CURE chronic kidney disease program. Rick previously has talked about the All of Us type 2 diabetes exposomic study. That data is now available in the All of Us research portal to access. Our commitment to data sharing. So those 55 studies all will be available on the HERE data center webpage. 39 of them are already public. The remaining 16 are still under embargo. That repository has supported 700 research users, and more than 3,000 file downloads have been supported there. The capability or the contributions of HERE have gone beyond those studied projects. We included a significant effort on advancing exposomic analyses. A lot of this has been taken up by the NEXUS program to really disseminate these new methods, multi-class methods for targeted analysis, expanded untargeted analysis capabilities, software and flows. And all told, NIE HERE has supported more than 500 publications with more than 12,000 citations. We did a newsletter program. We did a 21-episode exposomic webinar series. All of that information is still available and out there for those who are interested. With that, I am basically right on time, but I think we can take a couple minutes for questions if you have questions, and I will stop sharing so I can see you all. Any questions, comments, feedback? Daryl.
Thank you very much, David. I noticed that I was just wondering if you could upload your slides. There's so much information, so much great information that, so we appreciate that. But now I did have one question pertaining to the number of outstanding concept clearances that we heard over the last maybe 3 council meetings. And, I wasn't able to extrapolate from your projections what might be left for that. Can you give us an update, if you could, on that, or are you not able to do that?
So I think it's an open secret of the effort to reduce the number of NOFOs across NIH, and I think many of those concepts that you have previously seen will not appear in the form of a request for application, but rather where you will see them is in a highlighted topic.
That's what I was hoping.
So we are still committed to doing those activities. I think the way that we do them is going to be slightly different going forward. And I see Steph has her hand raised. The one NOFO.
David, I bet $100 you can probably guess what I'm going to ask.
the one NOFO that we are very much optimistic that it will be published soon. It's still under review, but our announcement for the fiscal 27 competition of the Superfund Research Program, P42s, that NOFO has left our hands. It is still under review at higher levels of the government. We are optimistic that it will be released in time for us to still be able to issue those awards in fiscal 27. If it is delayed enough that we're unable to issue those awards in 27, we've got a plan B, which I won't discuss publicly, but we have a plan B.
Okay, thank you. I appreciate it.
Okay, and I will try to get on Liam's calendar coming up. Okay. All right, thank you, David. Yeah.
Yes, as always, we encourage any applicants that are interested, reach out to your program officer. Have a conversation about your ideas and what might be the most appropriate mechanism to support that. If you don't know who to call me, and I'm happy to get you in touch with a program officer. We remain. One of the things that I think is special about NIEHS is our commitment to customer service and to working with our grantees, having a relationship with them, knowing who's working on what, and helping them get access to what they need to be successful. If there are no other questions or comments from council, we will be posting my slides, at least to the ECB. I think they also get posted to the council website. There's nothing super sensitive in there that can't be shared. With that, I think then we are set for a break. And so we will still plan on coming back on time at 11:30 to have a call to order and have Dr. Reed, the acting director of CSR, presenting on some of the new changes at CSR and have some discussion about the strategies for enhancing peer review. So with that, I think we'll break for 12 minutes and reconvene promptly at 11:30. Bye, everybody.